Assalamu alaikum dear students. Right now I want to discuss regarding how to store and quantify the nucleic acids. So, Alhamdulillah, we are done with uh, the methodologies and protocols for isolation of DNA from prokaryotic cells and the eukaryotic cells, including plants and animal cells. And we have seen that animal cells are far more easier to deal with when it comes to isolation of DNA as compared to the plant and the bacterial cells, because in plant and the bacterial cells we have to deal with the cell walls. In plant cells there is cellulose cell wall, whereas in bacterial cells there is peptidoglycan cell wall present. So once we get hold of the DNA, next thing is to store it. So we can either store the DNA in TE buffer, which is trace EDTA buffer, at 4 degrees centigrade for weeks, or we can store it at minus 20 degrees centigrade to minus 80 degrees centigrade, which is also known as ultra freezer, for long terms, for years we can, or for decades we can store it. Another excellent way to store the DNA for longer time period is to store them in lyophilized form, in which all the moisture is removed under vacuum, and then we turn the DNA into dry flakes. Those flakes can be kept for very long time periods. We can also keep the DNA for long-term storage suspended in precipitated form in the alcohol of the choice. It could either be ethanol or isopropanol, as we have discussed earlier. But if we want to work with that DNA for within months, then we can also use ultra pure water for its resuspension purposes. Like the hydrating agent can either be the TE buffer, which is tris EDTA, or we can use the ultra nano pure water, which is the double distilled water. You can also get it from a brand known as Gipco. Um, we that is the one which we use in our lab. When it comes to the RNA storage, we can store the RNA in ultra pure water, which is RNase free, at minus 80, minus 70 to minus 80 degrees centigrade for some time. We cannot store RNA for very longer times because it degrades quickly, because of its intrinsic property of having oxygen at carbon number 2. That is why it is more prone to hydrolytic species attack, and it breaks easily. Then it is also important to know that what is the expected nucleic acid yield. For that, you can see the formula at the bottom of the slide, where you can see if the cells, the number of the cell is 1 into 10 raised to power 7, then the expected DNA coming out from each cell is 6 picogram of DNA. Remember that we are talking about a human diploid cell here, and it is very sensible to calculate for 80% yield, because we cannot aim for 100% of the yield because of the human or the experimental errors involved. Anyhow, when you will calculate this whole formula, you will figure out that from 10 raised to power 7 cells, which are human and diploid cells, we can isolate 48 milligram of DNA. Here you can see we can quantify the DNA using UV spectrophotometer, which you have seen in the general lab, and you also know that that we can make use of the quartz cuvette for this measurement. You have also seen the nano drop spectrophotometer, where you can only use 1 microliter of the DNA for the quantification purposes. So if you are getting an OD of 1 and you are using the sample, double stranded DNA sample, this means that you got 50 microgram per ml DNA, double stranded DNA in that solution. OD of 1 for single stranded DNA means 33 microgram per ml of single stranded DNA. If you are calculating for RNA, then OD of 1 means 40 microgram per ml of RNA. And if you are calculating or analyzing for oligonucleotides, then it is 30 microgram per ml. You can also identify the total original concentration by multiplying the absorbance at 260 with the dilution factor and the values for each type of the nucleic acid. Like if you are calculating it for DNA, then you will add 50 microgram into the equation, and if you are calculating it for RNA, then you will put 40 instead of 50. So that is simple calculation. You can practice this sample calculation present at the end of the slides. The quality of the UV spectrophotometer can easily, you can use this UV spectrophotometer for qualitative analysis of our isolated DNA. And for that purpose, we will measure the UV, the absorbance at 260 nanometer as well as absorbance at 280. 260 nanometer gives us the quantity of the nucleic acids in the solution, and 280 gives us the quantity of proteins in the solution. So their ratio determines the measure of purity of our DNA or RNA. So upon dividing the two absorbance values which we got from our sample, if the values fall between 1.7 to 2, that indicates good DNA or RNA. You should be happy that you got a high quality DNA or RNA, your isolation protocol and your methodology was good. But if you are getting a ratio less than 1.7, that is somewhat alarming situation, which indicates that too much proteins or other phenolic contaminants are present in your purified along with your purified DNA, which means they will interfere with the PCRs later on. So it's time to repurify them. So what you are going to do, you are not going to throw this sample away. Rather, you can add phenol again to this aqueous DNA concentrate which you have got, and you can repeat the phenolic protein deproteinization steps as well as the chloroform deproteinization steps, followed by the ethanol or isopropanol precipitation and washing, etc. You can also check for the quality of agarose, quality of the DNA using agarose gel electrophoresis. But for this purpose, because we are checking quality of genomic DNA, we will be using 0.8% to 1% of the agarose gel. We won't go for the high resolution gels because their mesh size is very small and genomic DNA is larger in size. So only 0.8 to 1% gel is the suitable one for checking quality of the DNA, genomic DNA. And the high quality DNA won't travel very far on that gel. It will be present either stuck on in the well or just next to the well. But if you are getting a smear of the DNA and the DNA travels very far down the well, that indicates that your genomic DNA quality is not very good, and it had experienced shearing forces during isolation and it has broken down. Then we can also use are gel electrophoresis for quantitative analysis. For quantitative analysis, we can make use of the fluorescent dyes like ethidium bromide or cyber green. Ethidium bromide is the one which we have discussed earlier. Both of them intercalate between the bases. Ethidium bromide gives bright orange bands when exposed to UV, whereas cyber green, as the name indicates, it gives the green bands. But when it comes to the safety, ethidium bromide is not a very safe dye to use because of its mutagenicity. If unfortunately or if accidentally it spills on your skin, it is always advisable to wash immediately with plenty of water. And also, when you are dealing with ethidium bromide stained gels, always wear gloves. But whereas cyber green is a safe dye and you can easily use it without the fear of getting any mutagenic or toxicity effect. Here you can see the gels which have been stained with ethidium bromide and cyber green. For ethidium bromide, I will replace this gel with another gel where you can see the bright orange bands on the blue background, whereas the cyber green gives you the bright lemon yellow, lemonish green sort of bands. This slide tells us that the same DNA, when it is in double helical form, it will absorb less UV as compared to when it is single stranded. And by utilizing this fact, we can track when our sample DNA is double stranded or single stranded. Here I'm telling you another equation for melting temperature of the DNA. Earlier we have discussed that melting temperature is can be calculated using 2 multiplied by A plus T plus 4 multiplied by G plus C formula. Here you can see the TM can also be calculated using 69 degrees plus 0.41 multiplied by percent G plus percent C. Remember, here we are talking about the percentages and not the numbers. So if you have a DNA stretch of 10 nucleotides, and out of those 10 nucleotides 3 are G's, then you will put the percentage here, like 3 multiplied by 10, 3 divided by 10 multiplied by 100 will give you 30%. So this is the value which you will incorporate here for calculating the TM. In the bottom you can see the graph where half of the DNA, when it turns single stranded from double stranded form, that particular point is known as the melting temperature. You can see that you can rearrange this formula to figure out the unknown component. Like if you don't know the percentage of G or C in the DNA stretch, then you can rearrange it in a way कि आपके अपने you can put the ATM you can put the TM which you have calculated earlier, which has been told in question or in statement to you. Then you can utilize that TM for calculation purposes to figure out the percentages of G or C. And from G and C percentage, you can also calculate A and T percentage. Kindly practice it at home, and if you find it difficult, then you can come to my office and we can discuss it in person. In the last slides, you can see an effect known as hypochromism. Hypochromism indicates that the stacked bases absorb less UV as compared to the unstacked bases. As we have discussed earlier, that the double-stranded DNA absorbs less UV as compared to the single-stranded DNA. Why this is so? Because when the bases are unstacked, they are more exposed and they can readily absorb the UV. The same is true for the single-stranded DNA. When it is single-stranded, the bases are exposed, so they can absorb more UV as compared to when they are in the double-stranded form or when they are in the stacked form. So this particular phenomenon where we are talking about the stacked bases is known as hypochromism. You can also track the melting of nucleic acids very easily, followed by monitoring of their absorption at 260 nanometer. In the graph present on the same slide, you can see that the GC content also affect the melting temperature. Suppose we have 1000 base pair long DNA fragment, three of them, and one of them has 35% GC content, then its melting temperature will be around 65 degrees centigrade. But if we have the same length DNA, the 1000 nucleotide long DNA, but for that, if its GC content is 66%, then its TM will be 79 degrees centigrade. By the way, this is the genuine and the true data coming from Pseudomonas aeruginosa. And in the last, it is the AT-rich regions which melt first because they have two hydrogen bonds present between them, whereas GC are more resistant to melting because of their three hydrogen bonds. We can also calculate how much DNA can we recover. As I have told you earlier, that a human diploid cell contain approximately 6 picogram of DNA, and in an adult person, the average white blood cells, because they are the nucleated ones, in 1 ml of blood is 5 to 10 million cells. So theoretically, we can recover 30 to 60 nanogram of DNA per microliter of the blood, and that will become 30 to 60 milligrams of DNA per ml of the blood. But the real question is, how much DNA do we need? And as the techniques are getting advanced day by day, the less DNA amount is needed for each application. Like for a regular PCR reaction, we only need 1 nanogram of DNA. And there are many commercially available kits which are even sensitive below 1 nanogram of DNA. They only call for 100 to 250 picograms of DNA for a successful PCR reaction. With this, we are done with today's talk. I will send you the worksheets for calculation purposes. Best of luck. Thank you.